Good morning, and I'm looking forward to moderating this panel here. My name is Andrea Machlet. I'm with Bayer and the VP for Pharmacovigilance and Benefit Risk Management. And we have three distinguished scientists and clinicians here, and I look forward to the discussion. I liked how you ended on a very optimistic note, Dr. Hilt, with your talk, and that there's hope that so many modalities are in research and so many people making an effort also translating that into the clinic. That said, it's a huge step for a research organization to plan for a clinical development program. And we are here to discuss a bit more, have an opportunity to not only hear about approaches in Alzheimer's disease, but also in other neurodegenerative diseases, hopefully, and dive a little deeper in understanding what are the approaches, what works, what should be considered, and maybe I'm curious to maybe also hear about your personal experiences, what were your successes and your failures. And while we are going along and asking these questions, maybe you can also say a few words about yourself, your background and your involvement. And let me start with Dr. Hang. So going into clinical development, what comes to mind? What would you like to also maybe relate to people in the research space planning for that?
Yeah, so hi, everybody. Can you hear me? All right, cool. So I'm Serena, and I am a neurologist by training. I've been doing clinical development for about 15 years. Prior to that, I was in academics. And so during this time, I've worked at bigger companies. I've also worked at pretty small companies, as small as 14 people. And so I think, what drives clinical development, no matter what size organization you're in, is actually the fundamentals of good science. So, for a clinician looking at compounds, what do I want to take into the clinic? I mean, we have to be convinced about the science that it actually does stand a chance to benefit patients. Because otherwise, I feel very strongly about this, is that if we actually bring stuff in that actually doesn't have a lot of scientific chops to it, you're wasting patients' lives, you're wasting their chances of maybe enrolling in something else that might, benefit either them or the field more. So I think it's actually a very big responsibility that you're asking people to do a lot, especially when you're asking them to do first-in-human trials. They're taking on a lot of risks. No one knows, despite all the tox studies that you do. Animals are not human. Things happen. So you're actually asking a lot of from patients. So I think that's actually is sort of like the number one thing when we're working on a program. How do we want to do we want to approach clinic? And then afterwards, you want to think about what is the best way of doing it, because I think we're I don't know if we're going to touch on that or not, but there are different ways of engaging patients, and for a phase 1 clinical trial, the burden on patients and their families are tremendous. So is there anything that you can do to actually make it easier, but not sacrificing the scientific integrity of your phase 1 trial, which is an experiment? You still have to actually get to the points of answering key scientific questions, because that's your value inflection point. You have to know after your phase 1 trial, are you going to move forward? So you have to have clear answers. So how do you actually balance the two? That's actually things that I find to be fascinating, but immensely important.
Thank you. And I guess we can talk about the practicalities of doing the studies at a later stage, maybe. But I was curious, also following up on the previous keynote. So what's your thinking about to actually demonstrate your drug or modality is actually in the biological pathway of the disease. So, Dr. Hild, what do you think? How can we be sure that the understanding of the biological pathway is really underlying the clinical research program?
So, we say we want to understand the mechanisms of disease, but, it's still not entirely clear how insulin works, okay? And it's been used as a therapeutic for decades and decades. So, and the FDA doesn't require you actually to understand how your drug works. They require it's safe and effective. The number of times I've had the FDA or the EMA or the PDMA or whoever ask me about a mechanistic question and a scientific question, I can count almost on one hand. So drug development is a trade. It's empirical. You have to have a good molecule. We're much better at picking good molecules now. I mean, molecules that fail for poor bioavailability, horrible kinetics, those are much less frequent now. Okay. Then in the preclinical model, when I was an academic neurologist and a scientist, I always thought, well, the degree of efficacy in the preclinical model is going to be predictive of the degree of efficacy in humans. No, it isn't, okay? What the preclinical models will tell you is that your drug is active. I almost have a reductionist view of it now. It's like it's positive or negative. It's active in the model. Then I need to determine what is the exposure level in the target tissue that's associated with activity. And then I need to try to figure out how I can measure that in patients. For example, we now typically do CSF levels, okay, after dosing, even in phase 1 trials, to determine is the free drug concentration in the brain being measured in the CSF, is it in the therapeutic potential range? That's one of the most important things to derive from an early stage trial, because in normal volunteers, you can't show your drug works, but if you can show you get an appropriate, potentially therapeutic exposure, then you can go to the next step. The next step is, do I have a biomarker indication that the drug is active? Sometimes you can find that in normal volunteers. There's a great interest in Parkinson's disease, and LRRK2 is a target. I'm working on a separate project on that. And you can tell in normal volunteers whether or not your drug is actually inhibiting LRRK2. And you can measure the CSF concentration. So then you can say, aha, okay, I know the drug's active. I know it's getting the target tissue. I can then go into a clinical trial in patients. Okay, and then so I think these are some of the ways we think about drug development that when I first started doing this 30 years ago, they weren't front and center. We used a much more empirical approach. Let's give it to patients and see if it works. That's no longer the case. It's much more scientifically driven. It's still a challenging task, but I think we've made a lot of advances in understanding how to do this in a more rational way.
Thank you. And you remarked on biomarkers. I think that's a very good field for further exploration. And first, in human trial, we'll need to demonstrate safety and tolerability, but maybe also pick up on early signals for efficacy. And having a biomarker to be able to do so would be wonderful for a program, but for diseases that may have also a long trajectory, how they worsen over time, it's not so straightforward as measuring blood pressure, I would say. So what are your thoughts about the different types of biomarkers, liquid and others? Professor, maybe you have some opinion here.
Sure. Oh, I have lots of opinions. So I think that just like, I'm an academic. I've always been an academic, still am an academic, probably going to die an academic. But as a result, what I spend a lot of time thinking about is what are we not doing? Because we're really great at, once we understand what we are doing, I think there are lots and lots of people who know how to do it and do it well. So I focus a lot of time on what we're not doing. And in the realm of biomarkers, what I think about is how do they address that problem? And the reality is that, and we all know this, we do bias science. So if we say everything is based on the science that we do, then we have to really worry about the fact that we don't actually do the science in a way that is going to get us to our objectives. We talk a lot about precision medicine these days, but we're not doing the work that we need to in order to make precision science, actually, precision medicine, happen. And that's because we're leaving lots and lots and lots of people out, right? We do research that, and we know this. We know that we have biased samples, and that's because of the way we recruit them in. Mm-hmm. And I think where biomarkers have a very critical opportunity is to change that narrative. So if we start with fluid biomarkers, right? So right now, the big breakthrough is on the blood-based biomarkers. Why is that? Because generally, we know how to collect blood. When I say we, I mean everywhere. In the lowest of low income settings, they know how to collect blood. And there's such a richness in that blood. Blood in its raw form, undifferentiated, can be measured and in many, many different ways across many, many different diseases or risk factors. We talked about inflammation. We can measure inflammatory markers. We can measure all sorts of other cardiovascular factors. And now we can measure AD biomarkers. We got the genetics, genomics in there. So blood is so powerful as a way in which to do the kind of science that we want to try to do, which to me is inclusive science. And the other area that I'm very excited about and interested in is digital. Now, when you say the word digital, people have started to, well, first of all, the concept of digital and biomarker as a term, that's actually only a recent phenomenon. Because as someone who tried to put those two words together years ago, I used to get reprimanded that doesn't work. That's not a real concept. So today, everybody uses the term digital biomarkers whenever you take a digital device and you measure something with that. And they call it a digital biomarker. Well, it's not a digital biomarker, it's a digital measure. And just like we think about how do we validate imaging biomarkers, fluid biomarkers, et cetera, you don't just measure something from blood and say it's a biomarker. You have to go through a validation process. We got to do the same thing with digital. And so I'm excited about the prospects of digital biomarkers. I don't think we're there yet in terms of how to get there. But why is it so important? It goes back to what are we trying to do? We're trying to reach people. We're trying to reach all people. And it turns out that the most penetrating technology in the world is a smartphone. I actually wonder, is there anybody in this room that doesn't have a smartphone? If so, raise your hand. Do you not have a smartphone? Right? Well, it turns out over 6 billion people in the world have a smartphone. And a smartphone is a computer in every person's hand. It has multiple sensors in it. Those are data collection instruments. So suddenly, we have the capacity to collect digital at a global scale. And if we can get sort of this idea of how do we get from digital measures to digital biomarkers, I think we're going to blow open the opportunities of science.
Thank you. And I encourage everyone to maybe look at some of the publications that speak about how to use a multisensory brain health assessment tool, so including voice and markers like that, where maybe I have not thought about it until recently, but they offer a lot of promise. And so very interested to hear also from the other panelists.
Can I make a comment? I completely agree with what you said. So I've done as a clinician, took care of patients and did, Parkinson's disease trials, for example, where it's motor movement and so forth. So Mr. Smith and Mrs. Smith comes in for their visit, a trial. Mr. Smith, I say, Mr. Smith, I want you to get up and walk down there to the end of the hall, turn around, come back as fast as you can. And Mrs. Smith is behind Mr. Smith saying there's no chance he's going to be able to do that. So Mr. Smith, I'm wearing a white coat, so Mr. Smith pops up, walks down to the end of the hall, comes back and sits down. And Mrs. Smith's jaw's on the floor saying, he can't do that at home. And then you think about it. I see them for a half an hour every 3 months, all right, in a clinical trial. The rest of the time, I have no idea what Mr. Smith is doing. But now, with digital monitors, we can actually measure Mr. Smith at home all 3 months, and we can see how he's actually doing in his house, going out to the grocery store, and it will revolutionize the way we do clinical trials. And the FDA is recognizing this and starting to think about, okay, can these be a substitute for actually the measures a doctor or a clinician would do when the patient comes in for a clinical trial visit? So I think it will change.
Yeah. So actually, just to add to that, recently I had a conversation with some Huntington's disease researchers, and they told me that like the FDA actually has a funding for advancing digital health measures to advance them into biomarkers. And not only that, so money is one thing, but the other thing that they do with these grants is that they actually have very frequent meetings with the investigators, so that both sides are sort of moving in a congruent direction, so that in the end, it's not like researchers come in, this is the best biomarker in the world, and the FDA is like, what are you talking about? So, I think there's actually a lot of promise, and people realize that, different groups need to, with different perspectives, need to work together. So I think on the digital health measures, that's a very promising front, and a lot of effort are being put into it.
There's a project underway now that the FDA is engaged in and supporting to actually, with Parkinson's disease, validate the digital outpatient measure against the standard measure for drug approval. And so I think maybe in our lifetime, we'll be able to see that might be the measure that we can use in a clinical trial, which is a more realistic measure of patient function.
Yeah, very interesting. I want to ask a follow-up question. So we heard about approaches in Alzheimer's disease and Parkinson's disease. You mentioned also Huntington's disease. So these are all different potential target indications and diseases that vary in their origin and cause of disease, but also in their course of disease, the trajectory and the speed of progression. Any insights from your experience as a clinical researcher or clinical trialist also to what to consider in terms of these differences? Common diseases, rare diseases, pediatric or geriatric populations? Please.
Yeah, so I guess now, feeding off of myself, I mentioned sort of how blood is very powerful because in its raw form, it's undifferentiated, and then you can use it to measure many, many different markers across many diseases. Turns out digital is the same thing. So I like to say digital is actually the new blood. So what happens is that when you collect all this digital data in its raw format, in its multi-sensor format, it actually holds the potential of being able to be used to track lots of different indicators, just like blood. So you can collect digital, you can store digital, and then you can analyze it. And you can analyze it thinking about different outcomes of interest. Same thing. So I do a lot around digital voice, as you mentioned. There's a richness in when you speak. It's a cognitively complex task. Also holds a lot of other behavioral indicators, such as mood indices. So from that single voice recording, depending on how you progress, if I've been collecting it longitudinally over time, I can then go back in time and I can reanalyze it and I can look for those indicators that's specific to whatever the pathways you are. And I actually did that. I started collecting digital voice recordings in 2005. I did this at the Framingham Heart Study. And I, at the time, had no idea that digital voice would become potentially a metric, a measure, actual data. So what happened was, when Siri came out, that's when I realized that wow, where digital voice is headed versus when I first started a few years earlier is completely game-changing. And so I started to recognize what the potential is here. So I started collecting this in 2005. My first publication, because that's how we measure in academics, my first publication, after years and years and years and years of telling people that this is of some value, was in 2017. The vast majority of my publications have been since 2020 and forward. And that's because all this science has now emerged that allows us to analyze digital voice. I happen to be doing it related to Alzheimer's disease because I've been collecting it since 2005. I have recordings on people when at baseline they were still cognitively attacked, and now they have progressed to MCI, to Alzheimer's disease. We even have pathological confirmation at autopsy. And so in that way, I've been able to do it AD, but I can do it for PD. I can do it for, we don't have very many ALS, but I can do it for them, frontotemporal dementia. I can do it for people with depression. I can do it people with cardiovascular disease. So I can, from that single voice recording, depending on the pathway of that person, I can look for those markers. And that's why I think digital is the new blood. The big difference is that it's a non-diluting resource. So when I've collected this, I can actually reanalyze it as many times as I want to whatever contemporary standards and never lose its value. And that's the
Thank you.
That's the opportunity.
Very interesting. Dr. Hang, what are your thoughts on that? You spoke in our previous conversations about how to pick up on a signal early on, even if it's an indicator, not something that's already established, and then develop rigorous strategies around how to validate and use it going forward. Can you share some example, maybe?
Yeah, so I've worked on larger indications, and I've now worked on a number of rare disease indications, some with heavy dementia components, some without. But one thing that I realize is that there's actually a lot of strategies that you can kind of borrow from these different neurodegenerative diseases. Also, the other thing that was actually makes a lot of sense to me is this approach of maybe like I'll use microglia as an example. You have a target looking at microglia to target Alzheimer's disease, and you start thinking about how do I actually enrich a patient population so that I can actually get to a biomarker read earlier, so that I can have a go, no-go decision pretty early. So, you can have actually like, different strategies, like using a genetic disease like ALSP, for example, which, you can actually use it maybe just to look at your microglia markers to sort of give you a hint as to, am I actually even doing, what the drug is supposed to do? Does it have legs to move forward before I go into an Alzheimer's, indication where maybe the pathology, the biology is a bit more diverse, so you may not be able to get, as clear of a signal. So just think about, enrichment strategy in your early phase so that you can actually make decisions early on and have more confidence on at least the biological aspect by using biomarkers.
Thank you very much. I want to switch gear a little bit and talk about the fact that patients are in the center of the interest, and this is why we are all doing our work here. Nevertheless, being in a clinical trial can be a burden, and there are a lot of hurdles, as we heard earlier today, from the speakers. So what's your thinking about how to reduce patient burden, how to be more inclusive? What could be done to make it easier for people to agree participating in a clinical trial? Dr. Hild, what are you thinking?
Yeah. So I was an academic neurologist, and a scientist. And so when you're a scientist, you measure everything, okay? Hundreds of things, no problem. So you get to clinical trial. My first trials I designed, they were too complicated, okay? And there were too much patient burden. And so I've learned over time that less is more. You need to try to hone down what you're doing in a clinical trial, try to think about it from the patient's perspective. The other thing I learned is that when I have what I think is an appropriate design, I go to a clinical site and I say, let's walk through a patient visit, okay? And then I realize, well, the patient can't be in two places at once. And 5 hours of testing, the poor patient's going to be exhausted. And so try to make this much more practical, focused. I think with things that you were talking about speech recognition, smartphone utilization, or remote assessments, a lot of these things can be done remotely. You can interview patients, you can do the CDR sum of boxes remotely. That's done routinely now. And that's the primary endpoint in an Alzheimer's disease trial. So I may be able to get the CDR sum of boxes at the 3-month visit instead of having the patient come to the clinic. Maybe I can go to the patient and do the interview on a Zoom call and get a perfectly valid assessment of whether the patient, what's the level of the patient's function by doing this guided interview. So I think that it's very important to think about it from the patient's perspective, reduce the burden. I think we take too many blood samples in clinical trials. Every time the patient shows up, we bleed them. And so, is that really necessary? So I think, again, approaching it from the patient's perspective, putting yourself in the patient's shoes, thinking about what's the burden here for the patient is very important. And then facilitating the patient's visit. The most precious thing in a clinical trial is the patient. If you have to send a car to pick them up, to drive them to the clinic, so what? It's a modest expense, and it's going to really make their life easier on that day. Instead of them having to drive and park and be stressed, you can say, look, we'll pick you up at 8 o'clock and we'll have the driver take you home at noontime. And with the cost of clinical trials, the per patient costs in a clinical trial, an 18-month Alzheimer's trial, it could be $100,000 or more per patient. Okay, so spending $50, getting the patient and taking them home is trivial. And if it helps them stay in the trial, feel like it's easier on them, I think it's a good investment. I mean, that's a few thoughts anyway.
Very good. Any other thoughts from
Yeah, I'll
Emma?
pass to yours. I completely agree with, trying to think about moving trials into hybrid trials is a very hot topic right now. Now, with phase 1 trial, first in human, that's a little hard because you need to watch them. They need to come in, and depending on what molecule you're using, the inpatient monitoring time can be a couple days. So there are certain things that are unavoidable. So that's actually where the feasibility support comes in. So I'm working on a pediatric epilepsy trial. So I'm thinking about, I need to bring the child in. The parent comes in, but they have other children at home. What am I going to do with them, or what are they going to do with them? So actually, in terms of the trial planning, this is actually where having a patient ad board is very helpful. Because they are going to tell you, hey, this is not going to work. Even a hotel is not going to work. You have to pay for a babysitter, maybe. Or so you just have to actually understand, from the participant standpoint, what is necessary, and then plan accordingly. And then also from a hybrid trial standpoint, there are actually a lot of people thinking about very smart things to do. Like, for example, NFL. Now there's actually, a movement where they're not bleeding people anymore. They're actually putting it on a, finger stick on a card, and the patient sends them back, right? So there's actually things that we need to just look for strategies to actually help people complete the trial. It's a huge undertaking on their time and on their family's time. So I think we just need to be as thoughtful as possible.
I actually like to take a step back, which is thinking about the participant, well, first, that they're a person before they're a patient, and thinking about what's in it for them, and starting to address that. Before you even start to recruit them in a trial, why is this going to be meaningful for them and for potentially their family members? So thinking about sort of this idea that before you even start your trial, think about the communities that you're trying to reach and start to engage with them and try to understand what's important to them and start giving them what they need. And then as you start to recruit them in their trials, there's lots of things that all of you have suggested. I certainly think moving as much of it away from having to come in is going to be very critical if you're going to be more inclusive. And then the other thing is, what are you going to give back afterwards? I think at the end of the day, it's this idea that you should be establishing some sort of much more longer-term relationship so that you're thinking about the value proposition from their perspective and not just from your perspective. So for instance, one of the things that happens is they go through clinical trials, then it's done, then what? I mean, their interest in their health, their brain health in particular, continues to go forward. And what can we do as a community to make sure that we build those resources so as they finish up the trials, they have elsewhere to go and continue on thinking about and caring about their brain health? So I think that we need to be thinking about this as a before, during, and after prospect. And thinking more creatively about how do we start to address that so that it's not just a one-way relationship, but it's a bi-directional one.
Can I add something to the relationship? What are they going to get back? This is actually one lesson that I learned is transparency to the extent that you can. At the end of the trial, tell people what happened. Patients get very frustrated to say that I actually find out the trial results from a press release. That's very disappointing. And we have an obligation to our patients and the trial participants that we disclose to them, whatever we can. There are people who actually don't get their assignment, like were you on placebo, were you on active drug? It makes a huge difference because if you are on placebo, you can immediately participate in another trial. But if you don't tell them, then they can't, because then you are actually, the next trial, the people are going to assume that they were on active drug. So it blocks them out for like 6 months or maybe even longer. So that's hugely unfair. So I think as trialists, we need to be thinking about these things, and this is our responsibility.
Also, many trials have them, but open label extensions are important. So for example, if the patient is randomized to a placebo or active during the trial period, if they know that they will get the active drug for some period of time to see if it might be able to help them, that's very important. And many sponsors companies do that, but sometimes in smaller companies, there's a budget crunch and this isn't done. But it's not only good for the patient, it's actually good for the trial because it encourages patients to enroll in the trial. So it's good for everyone.
Thank you. I want to follow up on what you just said, and long-term follow-up studies, but also abilities to accelerate the clinical development. I think that would be in everybody's interest. We heard earlier, so trials need to be conducted slim and fast, ideally. So what is your thinking about accelerated pathways and pursuing an approach where you can demonstrate efficacy potentially based on surrogate markers and follow up with endpoints later on? So any experiences you want to share, or maybe also any pitfalls that people should consider in such an approach?
So the FDA's approach, I shouldn't speak for the FDA, but in my interactions with them, there are biomarkers that show your drug maybe has some physiologic action, then there may be clinical biomarkers, and then there's a surrogate biomarker. So a surrogate biomarker in the FDA, in the regulatory sense of the definition of the word, fully captures the clinical benefit that you see. So it can be used instead of a clinical endpoint, okay? So the FDA, for example, has said that amyloid imaging can be a surrogate biomarker for expedited approval of an anti-amyloid antibody followed up by clinical data over time. And so that can facilitate an earlier approval, let's say, of an anti-amyloid antibody. Sometimes that may be, some can question the exact correlation between the benefit and the surrogate biomarker. And it's very hard to achieve a surrogate biomarker designation from the FDA. They don't give them out left and right. It's very, very difficult. But I think from my perspective as a drug developer, the more important even than that is a biomarker that is intermediate between a test tube biomarker that I can measure my drug is having a physiologic action and then a clinical endpoint. Is there something intermediate, like some normalizing effect on brain activity? It's really very valuable because then I know a particular dose or exposure, it's headed in the right direction. I'm having some impact which seems to be beneficial on brain activity. And those are the point of focus. People are trying to identify those. There's some available, but there aren't enough. And because you think about a disease-modifying trial, the lecanemab trial, 1,700 patients over 18 months, 5, 6 MRI scans, PET scans. I mean, it's a huge endeavor to show a drug works. If you had something that was intermediate, shorter, smaller numbers, it could really help drug development and get the drugs through the pipeline to patients more rapidly, I think.
I think one of the things, though, that you have to be very careful if you make it sort of much more biologically based indicator is the fact that just because you have the pathology doesn't mean you'll have clinically expressed disease. And I think that that's really important, and that goes back to the bias of our science. Just remembering that we have very biased data in which we're making lots and lots of presumptions. And here's one of the things we don't know. We don't know how many people in the general population have amyloid and tau in their brains. What we do know from neuropathological studies is most people do. It is very hard to find someone at autopsy with no pathology in their brains. So given the fact that we actually don't know, I think it becomes very difficult to start with that premise because you always have to think about what are you making your presumptions on? What's the data, right? At the end of the day, we have to think, what does the science tell us? But also, what is the science not telling us because we haven't collected the right data? So I worry a lot about the fact that we're moving more and more to this biological basis of disease when I know a certain proportion, I don't even know what proportion that is, won't clinically express. And I certainly don't know that once I get into much more racially, ethnically diverse populations. So I think that what you have to do, it's never one size fits all, but one of the things you have to think about is how do you measure then that clinically meaningful indicator that suggests that this is something that's going to progress to disease, right? And that's where I think that our tools, one of the things we have to realize is our outcome measures, our clinically meaningful outcome measures like the CDR sum of boxes, they've been around for decades. Like we spend so much money worrying about how do we detect the pathology of this disease earlier, and earlier. Our outcome of interest is supposed to be about clinically meaningfulness, and we make no investment in changing those up. We decide that, sure, we're going to continue to use these decades-old measures that were developed well before the concepts of MCI, preclinical, prodromal, asymptomatic. Well, I think that, in fact, if we focus a lot on thinking about how do we detect those early symptoms much earlier on, what we consider asymptomatic very well may not be asymptomatic. How do we know that? We know that because if you ask someone, when they come in, how long have you been noticing symptoms? They'll tell you they've been noticing it for quite a long time. And their family members will do that. They're not going around testing them. They're observing these very subtle changes in behavior, and they're observing it over long periods of time. We're just not doing anything to measure them.
Well, thank you for that viewpoint. So I think I want to ask one last question to the panelists, and then maybe we allow for some questions from the audience also. Please.
I actually want to expand on the accelerated approval point, because I think as drug developers, we're always aiming for accelerated approval. And in certain cases, it is appropriate. In certain cases, I think from a regulator standpoint, the implications to society is too large. That if we get it wrong, we can potentially, harm people. So that certainly has, looking at how the FDA has been looking at accelerated approval, you can see that it seems like it's much easier in rare diseases. Because if they get it wrong, it's a few, thousand people, a few, like tens of thousands of people, but it's not millions. So I think their attitude in terms of granting accelerated approval would be quite different. In the rare disease space, the FDA has over and over again in the last few years basically telling us, come and talk to us. If you give us a coherent story, essentially, with the totality of data, then we can have a conversation and see if that is a viable pathway, and also understand whether the data that you have generated is sufficient for an accelerated approval. So I think it's very promising in terms of this approach. But I think we also, on the drug developer side, we need to actually think through, what are the data that we have? And what is it telling us? So I think it's a pretty complicated story. I think we should always think about whether we can do it, and and identify opportunities to do it if it's appropriate, and if you have the support for it. And one thing that actually, this is personal opinion, but one thing that bothers me is that the FDA actually does not have the enforcement teeth or enforcement authority to pull something off the market. If they actually granted accelerated approval, and then later on they would say, we recommend. Yeah, oops. And so, there would be things hanging out there, the phase 3 trial, the confirmatory trial, either have not been done in 10 years, which is kind of ridiculous, or they've been done and it actually failed. Yeah. But the FDA cannot pull things right away. And patients don't necessarily know, some clinicians don't necessarily know, they keep prescribing things. So I think there's actually a lot of implications in terms of accelerated approval that we need to think through. But when the opportunity is right, when the data support it, I think it's actually a great pathway so that we can get promising therapy to patients.
Thank you. And you made a very good point in long-term efficacy. I think what's also important is the considerations of benefit-risk, more holistically thinking about safety, but also including what else a patient may have as options, what's the unmet medical need, and are you potentially also shortcutting pathways to alternative treatments. So all of that should be considered when devising a clinical program. So I think maybe we are at the point where we could also allow some questions from the audience.
Particularly if they're easy questions.
So feel free to use the microphone in the middle for questions.
Please come forward to the mic.
Maybe I'll try to just speak loud.
Yeah, but we have online, they will see you.
Oh, right. Thank you so much for the wonderful discussion today. Alzheimer and dementia, truly global diseases, and I think in the morning we spent a lot of time talking about mostly what being done around the country, within the U.S. Having taken care of my mom who suffered from this, it's very sensitive topic, and one of the really reasons I work in oncology, but I really wanted to hear what is the latest and greatest really in this field. I wonder if you could please share your thoughts and insights on the global environment, what the challenges are and possibly what the opportunities are in terms of really the ecosystem around neurodegenerative disorders research and clinical trials and so on and so forth.
Mm-hmm. Yeah, I've actually spent a long time thinking about this from a global perspective. It's actually why I've kind of pivoted in terms of how I do. So part of the reason I'm now very centered on that smartphone is because it turns out that there aren't actually a lot of neurologists and neuropsychologists when you start to go outside of the US and Western Europe, right? Even in the Asian countries, I mean, I've spent a fair amount of time in China. I think at one point they had 300 neurologists. It's a country of 1.2 billion people. There are no PhD programs in neuropsychology in most of East Asia, by the way. Actually, there are none. There are versions of them, but there are no real clinical neuropsych programs. So how do you solve that? How do you solve that? And we have sort of in the African countries where they're now rapidly aging as well. So this is becoming a global pandemic, but we don't actually have the infrastructure in place to look at this disease across the board. So that's why I'm focused on sort of two things, digital and blood, right? So we've talked about sort of the blood-based biomarkers. Besides the AD ones, there's other ones that are sort of under development like TDP-43, et cetera. And if you sort of pair those two together, you suddenly have a tool that can be administered almost anywhere and potentially by anyone, right? The idea with digital is that it can be self-administered. So you don't need a clinician, you don't need an expert. So that's what I think a lot about, is how do we develop and do this work without having the need for expertise and infrastructure in place, because they don't exist in many parts of the world. And so that's been my solution.
Yeah, I mean, I'm doing a trial in Alzheimer's disease with clinical diagnosis, and then we confirm it with a plasma p-tau181. So,
Right.
And then at the CTAD Congress in Spain week before last, it's now one of the labs working on p-tau markers now shown you can do it with a pinprick on a card.
Right.
And so, you can basically have clinical diagnosis, the pinprick, the patient can send the card in and you can confirm. Now you have to be careful to make sure that it's really Alzheimer's disease, but that's vastly different than a $5,000 PET scan, which doesn't exist in many countries, or trying to do a lumbar puncture, and then having the complications and discomfort, the expense of that. So that's going to transform the trials, but it's also going to transform care of patients, particularly if some of these antibodies to amyloid can be developed that can be given every 6 months or don't have as many side effects. So then you could start to see that that's actually a more feasible type of treatment scenario in many different countries.
Yeah, so actually thinking about, how we normally get approvals. So, with the trials, we need to have, representative population that we actually fail quite miserably. But let's say we achieve that. There is actually always the question from regulatory authorities in other countries that basically be like, what is the biological basis that if your trial is done primarily in Caucasians, that it's going to work for, let's say, some Asian population. And certain countries would actually have the requirement that you have to do certain things in their respective countries. And so I think for us, in terms of thinking about, the biomarkers, the variability in different populations, that's actually one way to support that maybe biologically we're not so different. And that could be a basis to actually give people confidence that, whatever work in the big trial, it's probably going to work in other population as well. So I think that's something that we need to actually keep paying attention to and advancing.
Thank you. A lot of challenges, and it sounds like also a lot of opportunities. Thanks.
Are there any other questions?
I want to ask a question. So in your talk, you talked about how when you get diagnosed with cancer, you get a lot more information about the type of cancer you have.
Right. What the appropriate therapy is for you.
What's the appropriate therapy for you? what opportunities, sort of what's on the horizon for neurodegeneration in terms of us understanding which patients we treat with what?
Well, we're at a very early stage there, I have to say. But some of the data, again, at this Alzheimer's conference, suggests that, you know, these anti-amyloid antibody therapies, once you have this explosion or rapid increase in tau expression, they're probably not appropriate or don't work as well. So we have to think of other therapies for that stage of the patient, and we shouldn't be exposing those patients to the risks of getting those anti-amyloid antibodies perhaps too late in the disease. We should focus on tau or other pathways. So as our understanding of the diseases evolve, then I think we'll be more specific. And the same Parkinson's disease, there are many genetic causes now of Parkinson's disease. So is it one size fits all for a therapy, or do we really should be doing more genotyping and segmenting the population and matching the therapy to the particular variation of Parkinson's disease that the patient has on a molecular basis? Then one final example is the one I mentioned about FTD. You know, there are 5 different phenotypes, can be caused by the same mutation. We shouldn't probably be treating them with different disease drugs. We should try to focus on the mutation, what's the biology of that mutation, and then qualify the patients for trials and for therapy based on the biology. I mean, cancer studies now have, you can have patients coming into an oncology study with different cancers. If they have that kinase mutation, that's the most important thing. You know.
But I would also say that the other thing we need to think a lot about is prevention, right? So Alzheimer's disease is multi-etiological, right? We know that there are multiple different pathways. So for instance, we know the cardiovascular one, right? The vascular pathway. So you can start thinking about treatment for risk for disease much earlier on with other kinds of treatments, right? So that's where you treat hypertension, you treat diabetes, et cetera. So I think that it's also about figuring out what pathway are you on and thinking about how do we identify those risks much earlier and treat that before we get to the stage where we're actually treating the disease itself.
Please.
Hi. I think that was an interesting segue into a question I wonder about as we think about these digital signatures and their accessibility. Do you think much about, or I'm sure you do, about whether or not these could not only be useful for us as drug developers and for the patients, but whether these could over time be utilized as or identify pre-existing conditions in the broader patient population where everyone has, you know, billions of people have these phones. So I wonder what you think about that risk-benefit scenario.
Sure. I mean, in my ideal, I think about not precision medicine, I think about precision brain health. And if we think about, for instance, Alzheimer's disease, dementia, basically any chronic disease, right? Those are insidious in onset. And we now think about dementia as a life course disease, where we know some of the seeds of risk are sowed very early, as in childhood, right? So to me, really, if we're really thinking about what's the pathway from an epidemiologic public health standpoint, we should be thinking about how do we use these digital tools, right? There's 35 billion IoT connected devices by 2025. So everybody has these. And so what you're doing is you're actually collecting a lot of the information across your lifespan that's actually informing where you are along this pathway. And again, I think that prevention should be high on our list of what we need to be focusing on. So I like to think a lot about precision brain health rather than precision medicine. And I think of precision medicine is something that happens once the trajectory of precision brain health goes off in one direction. Mm-hmm. So I do think that there's lots of opportunity in which these digital tools will allow us to optimize our brain health throughout our lifetime.
Just don't let your insurance company find out, though.
Yeah, I was actually going to say about, like while we're developing all of these data sets, technologies, and so forth, I think simultaneously we need to be thinking about the privacy issue. Yeah. What if, like our laws are not, really catching up with, our knowledge of genetics and, what are the risk factors and so forth. And like you said, if they get it and we're not actually, we haven't thought through the issues of privacy, they can deny you coverage. And, it can be misused. I'm just being completely paranoid here. But, there is actually a lot of opportunity to misuse. So while it's actually very promising, and I think there's, a lot of interest, especially, for general health, if we can actually do better at prevention, maybe we won't have so much dementia when we're, older. So yeah, but we actually also need to think through the other aspects of, what could go wrong.
Yeah. Thank you very much for bringing up the aspect also of data privacy and security of data. I think that's very pertinent and maybe the topic of another panel. But people think about also what to do with all these integrated evidence generation strategies that involve digital biomarkers from the blood. And yeah, so do we have time for one more question? If not here in the room, maybe we have anyone from the online audience?
Not yet.
Okay. So, please, yeah. Could you come to the microphone, please?
I'm curious about the patients with FTD that you mentioned, where the same mutation is leading to 5 different phenotypes. What is the reason for this?
I hope you're going to give us the answer. I mean, the fact is that if you have a mutation, this polymorphism and this mutation in this gene, well, guess what? You have billions of other bases that can be mutated. And so clearly, and then you have your life experience, the toxicities you've been exposed to. I mean, it's just very complicated. I mean, every patient actually is an individualistic patient. We have to group them into groups in order to cope with it in medicine, but in reality, they're all unique. So I think there are probably combinations of different genetic factors and then different life experience factors. In schizophrenia, identical twins, the concordance of schizophrenia is 50%, okay? It's not 100%, it's 50%. So, simplistically to me, that suggests that maybe it's 50% of it is genetic, but 50% is something else.
Very interesting. Thank you.
Yeah.